Um, Good afternoon, everybody, both those who are present here and who are online. So welcome to the departmental seminar of CBMS department. And it's a pleasure to welcome Dr. Soumya De. He is currently assistant professor in the School of Biosciences in IIT Kharagpur. Dr. De did his bachelor's and master's both from chemistry department of IIT Kharagpur, and then he did his PhD from Cornell University. He made a change in field. He moved from chemistry to biophysics, and he specialized in NMR spectroscopy, as we'll see today. So he completed his PhD in 2011 from Cornell University and had a postdoctoral stint in University of British Columbia in Canada. And he returned to India in 2015 and joined IIT Kharagpur and built his own group. And he's a structural biologist by interest, and he uses different biophysical techniques, characterization techniques to understand the structure function in biological systems. And he's going to tell us more about what he does. So welcome, Professor. Slide, sure.
Thanks for the generous introduction, and again, thank you for inviting me. Okay, I'll speak here.
That would be better if you want to move around.
So I'm audible. I'm audible now? Can you hear me now?
Yeah.
Okay. So, my name is Soumya De. I'm from School of Bioscience, IIT Kharagpur. So this is my research lab, research group. So I'm going to talk mostly about the work that was done by Snigdha and Soumya. So there are lots of collaborative projects also that we work on, but I'll not get time to talk about everyone's work. So as Dr. Chakraborty said that a lab is a structural biology lab, and we use NMR spectroscopy to look at protein structures. Right? And these are some of the protein structures that we solved in the last 5 years using NMR spectroscopy. This one is the N-terminal domain of a T-cell receptor. So these proteins are the ones which are on the surface of these T cells, which are immune cells, and they help in interacting with other cells. This is an enzyme which catalyzes a splicing reaction like that in RNA. So I will talk about this protein in particular, and this is an RNA binding protein. So you can see that we work with different types of proteins. So I'll talk about this enzyme as an example of a structured protein, and I will also talk about unstructured proteins, proteins which are intrinsically disordered. So what is an intrinsically disordered protein? So this is from an actual MD simulation of a protein that we work in our lab. This is a transcription factor. So it binds DNA and it regulates the transcription of genes, downstream genes. Right? So this folded region that you see, this the alpha helices, this binds the DNA, and then this region that is sort of flopping around, that's the disordered part. Right? And what we know is that this disordered part plays an important role in regulating the function of this protein. So one of the major work that we are doing now is trying to understand how something that is unstructured, has no structure, can help in the function of a protein. Right? So it turns out that proteins, they have this huge spectrum of structures. So there are proteins which are completely folded, like the intin enzyme I showed. So it's completely folded protein. And then there are proteins like this. So example will be P27, P59. These are completely disordered. So they do not have any structure. So they are just like this flopping polymer in solution. Okay. And then of course there are intermediate structures where proteins can have folded domain and small disordered regions, or folded domain and long disordered regions. So the protein that we are looking at looks something like this. It has a small folded domain of 60 residues and then long disordered region of almost 400 residues. So how do these intrinsically disordered regions in proteins help in protein function, right? Because they do not have any shape, they do not have any structure. So it turns out that there are two major ways by which they can help. One is by facilitating protein-protein interaction. So these disordered regions, they are actually hotspots for interacting with other proteins. So they can recruit other proteins, find other partner proteins, and bring them towards the whatever function they are doing. Like if it's a transcription factor, they will help in the formation of the transcription complex. Okay. So these type of proteins are found a lot in these protein interaction networks, and they tend to be these hub proteins, meaning that they interact with lots of different proteins, right? And these type of interactions are mediated by these disordered regions. Now, these disordered regions, they have certain motifs. So there are many types of motifs. I'll just talk about this one. So this is called SLIMs, or short linear motifs, which is something that we are looking at specifically. So these short linear motifs are 3 to 10 residues in long, and they interact with certain partner protein, right? So you can imagine that if there is this protein, this is the folded part, and this is disordered part. So it has one linear motif which binds to this protein, and another linear motif which binds to this protein. And then you can imagine maybe some more motifs. So this disordered part can facilitate interaction with 2-3 proteins simultaneously at the same time, right? And then of course it can help in the formation of a complex. So that's one way these disordered sequences can help in protein function. Another way that has been shown recently is that there are certain proteins, especially RNA binding proteins, they have these long disordered sequences which are called low complexity sequences, meaning that you will see that these sequences do not vary a lot. Very few type of amino acids keep on repeating themselves, like glycine, arginine, and then phenylalanine or tyrosine, right? So the same type of amino acids keep on repeating. So these are called low complexity sequences. And it turns out that this type of sequences, along with the RNA binding domain, they have this tendency to form liquid droplets. Okay. So it's basically, under microscope, it will look something like this. So this is very similar to oil droplet. If we take oil and water and mix it and then let it sit, you will see this small oil droplet. So that's the phase separation. So this type of phase separation can happen. And this type of phase separation is actually seen inside the cell. So it's not something that is something that people have seen in the lab in vitro. It is something that actually happens in vivo. So inside the cell, there are all these different types of membraneless structures, right? Like stress granules, P bodies. So these things were known long time back, but their physical characteristics was not known. So this is something that has been figured out in the last 10-15 years, right? So these are nothing but phase separation, and this phase separation are mostly driven by this type of proteins, mostly RNA binding proteins. So we started working with this particular protein, RBN3, in the last couple of years, and we think that it also forms phase separation. But anyway, I will not get time to talk about this, but that it's just to show that these disordered sequences can help in function in these two major ways. Okay, so I'll talk about mostly this part of the talk. So to look at these short linear motifs, we started working with transcription factors, and we started working with a particular family of transcription factors called the Hox transcription factors. So these are something that are present in that, and mostly a lot of work has been done in Drosophila. So in Drosophila, there are 8 proteins in the family, and in our case, there are 39 Hox proteins. And these transcription factors are involved in the formation of the body plan. So in the early development stages, these proteins actually dictate where the head will form, where the tail will form, where the limbs will come out, where the wings will form, right? For Drosophila and also for us. So it actually defines the body plan of an organism. And this is driven by the expression of these proteins in a timely fashion and also in a spatio-temporal manner, right? But that is what happens in the early development stage. But once the full-grown organism is there, then also these proteins have function. For example, these three proteins that I have highlighted, they are actually involved in the development of different types of blood cells. Okay, so we call them the hematopoietic cells, right? So, hematopoietic stem cell is the stem cell of the blood from which different blood cells are formed. So different lineages are formed, and these proteins actually play an important role in that. And it's not surprising that these proteins are involved in different forms of leukemia. So that's sort of the motivation of why we are looking at these human proteins, and to understand the basic principles, we are looking into this to Drosophila proteins. So how do these proteins look? They are actually quite big. They are roughly 400 amino acid long, 450 amino acid long. Only 60 residues are folded, which binds DNA. So structurally, it looks like this. And the remaining part is disordered. Okay. If we take the isolated DNA binding domain, in all this 39 plus 8 plus any organism that I take, they look exactly the same. Sequentially, they are very similar, and they bind to very similar DNA sequences. Okay, but the difference comes from this disordered part. So again, the question comes back that how this disordered part is actually influencing the function of these proteins. So it turns out that this disordered part, of course, helps in recruitment of other proteins. Some of them are also transcription factors. We started working with a particular partner protein that is known. So these are the proteins, and so SCR is a Hox protein, and extradentical is a partner protein. So the recruitment of this partner protein is driven by this disordered region, right? So how the partner protein is recruited by this disordered region, that is what defines the function of each of these proteins. This is just a slide I have put up here, so just to show that these, even in adults, these proteins actually have multiple functions. These three proteins are, they have been looked at a lot, and they have been involved, shown to play all these different roles in leukemia, right? So that's sort of the motivation of why we are looking into the human version of these proteins. Right. So how do we use NMR spectroscopy to look at this? Now we are looking at something which does not have a structure, right? So applying this, so as structural biologists, we are trained to look at structures, right? But then applying those principles to something that does not have any structure or three-dimensional structure is not going to work. So what is going to work is, instead of thinking in terms of structures, we have to think in terms of dynamics. And NMR spectroscopy is something which is a very versatile technique to look at protein dynamics. So this is sort of the range of dynamics that can happen in proteins. So this is femtoseconds, picosecond, nanosecond, microsecond, millisecond, and second. So very slow processes and very fast processes. And different processes that happen in these different time scales are listed down here. For example, very fast vibration motions happen in this femtosecond time scale. Right? Torsion angle dynamics, fast loop motions, they happen in this picosecond, nanosecond time scale. Ligand binding or substrate binding to enzymes or proteins, allostery, catalysis, they happen in this slow microsecond, millisecond time scale. And protein folding, unfolding will happen in this slow second time scale, millisecond to second time scale. And it turns out that in NMR spectroscopy, we actually have different experiments which can look into this different time scale. So again, one single experiment cannot cover the full range. So they have a particular window. For example, this type of experiment can look at these fast picosecond, nanosecond motions. And this type of experiments can look at the slow millisecond timescale motions. So depending on what type of dynamics we want to look at, we want to study, we choose different NMR experiments. So for disordered proteins, the motions are mostly in this fast time scale because there is no folded structure. So folding, unfolding is not going to happen. Allosteric, catalysis, ligand binding, these are all something that happens in folded structures. Right? What we are looking at is mostly this side chain rotation, torsion angle dynamics, and maybe we can think of fast loop motion. Okay, again, there will be strictly no loop, but what we see is a segmental motion. So I'll come to that. Now, all of these things happen in this fast picosecond, nanosecond time scale. So we have chosen this set of experiments to look at this type of time.
Just a quick clarification.
Yes.
Previous slide. So it seems that residual dipolar coupling has the largest range of picosecond to millisecond. So why not use that method for most of the problems?
Right.
We are more interested in that regime only.
Yes. So this you're right. Again, I mean, sometimes it's debatable how big it is, right? And the second thing is that the ease of doing those experiments. Now, residual dipolar coupling, it actually works for folded proteins. So in this case, what is done is the protein is put in an alignment media. So making the sample is also quite tricky. But then again, it works mostly for folded protein. I don't think I have seen any example of disordered proteins or sequences being studied by RBC. But I might be wrong. But this is something that is more conducible for. Okay, so this R1, R2, and NOE, these are actually experimental parameters, something that we can measure by experiment. So, but it turns out that these parameters are highly sensitive to this fast timescale motion. Right. So, what we are looking at using this is this NH bond vector. So, if I draw the structure of a small peptide, so this is a dipeptide, right? That's the peptide bond, and this is the NH. So this NH will belong to this particular amino acid. So for every amino acid, there will be a single NH, and we measure this R1, R2, NOE for each of these NH bond vectors. Okay. So for every residue, we get a particular parameter. So what we are plotting here is the sequence of the protein and whatever value we get for each of those amino acids. Right. So for each amino acid, we get R1, R2, and NOE, these three values. And these three values we can then use to measure the dynamics of that particular NH bond vector. So for folded, again, traditionally structural biology has been done for folded proteins, right? So for folded proteins, it's quite clear cut how to analyze this type of data. Right? So what people normally do is they look at the global tumbling motion of this protein, which will be same for all NH because it's a global motion, and that is extracted out, and then the local motions are extracted out. Okay, so that is a very well established method, but that type of analysis will not work for something which is disordered because there is no global tumbling motion. Right? We can imagine a global tumbling motion of a sphere, a protein which looks like a sphere, but when this is a disordered polymer, then there is no global tumbling motion. Everything is moving on its own. Right? So to look at that, it's much better to use something else. So what we do is we use a different type of analysis called the reduced spectral density mapping. Right? So what it does is we can look at this reduced spectral density mapping as so we measure this at 3 different frequencies. Okay, so how much for each amino acid, how much motion is present in that particular frequency. So this is something that again we have this motion which is happening in a particular time scale. If you want to get the frequency, you have to do a Fourier transform. So that is exactly what is done. So a correlation function is there. We do a Fourier transform of that to get the spectral density from that correlation function. and then we measure the spectral density at these 3 different frequencies. So this will be 0. 0 means very slow motions, and these are actually 2 fast motions. Right? For each amino acid, we see the contribution in these 3 different frequencies. What you will see is for the folded region, the contribution of the slow motion is very high, meaning that for the folded part, there are more slow motions and there are very less fast motions. And for the disordered part, there are very less slow motions and there are more fast motions, right? So which is intuitively seems right. So this is what we also see from the experimental data, and what you see here is just a blown up image of this disordered part. Right? When we blow this up, what we see is that in the disordered part, again, it's not flat. It actually goes up and down. So for some regions, like this blue one, the slow motions are less and the fast motions are more. And for these red ones, the slow motions are more and the fast motions are less. So it means that even in the disordered region, there is a variation in the flexibility, right? And so what we did was we actually combined all of this into one parameter. So we collected the 2 fast motions in the numerator and the slow motion in the denominator. So now what this will do is for residues, if this number goes up, it means that that particular residue is relatively more rigid, and if it goes down, it is flexible, right? So that's sort of the physical picture that we are trying to get, because what we have is this, and from that we want to get a physical picture of what is happening in this disordered part. So by doing that, what we see is that there are regions in the disordered part of the protein which are rigid, shown by these red bars, and there is a region which is highly flexible. Right? And then later on, we figure out that we don't have to go through this involved spectral density calculation, because there is a direct mapping to the original parameters that we have measured. So instead of calculating this, we can simply replace this J naught J omega n by this. So we replace that with this R1 R2 by 1 minus NOE. So instead of going through the spectral density calculation, we can simply get the same information from the parameters that we have measured. And you can see that the pattern is exactly the same. We see these residues with more rigidity and these residues with more flexibility. Right? So what does that mean? So what we are saying here is that these intrinsically disordered regions, even though they look like highly flexible, they are just flopping around. Not every residue is equally flexible. There are regions which are more flexible and there are regions which are less flexible. So if I think of my hand, there are these joints which are more flexible, and then there are these rigid parts which are less flexible. So it's something similar to that. And we say that these rigid parts are the ones which are hotspots for interaction with other proteins, because when they bind to these other proteins, they will come down to one conformation, right? So that's sort of you're decreasing the conformation, so you're losing conformational entropy. Here, the conformational loss in conformational entropy will be very high, which means that the binding will be very weak or maybe unfavorable. But in these cases, binding might be favorable because the loss in conformational entropy will be less. So to check that, we took this protein. So it's this protein, that's the folded part and that's the disordered part. So we took this protein and we titrated the partner protein, right, to see where this partner protein is binding. And now this is done without the presence of DNA. So we know how they interact in the presence of DNA, but here we did not add any DNA. We are just looking at protein-protein interaction. Okay. So when we did that titration, in NMR spectroscopy, we can actually pinpoint where exactly the interaction is happening, because each of the amino acids we can trace. So what we do is we label this protein with N15 isotope and we assign it. So we know each peak belongs to which amino acid. And then when we titrate this other protein, which is unlabeled, so it's invisible in the NMR spectrometer, the residues which are interacting, they actually shift like this. Okay. So when we see peak shifts like that, we know these are the residues which are interacting, and the residues which will not interact will not shift. They will stay right at the same place throughout the titration. And then we can map this change in chemical shift per residue. Okay, so that is what is plotted here. So this is all the residues in this protein, and this is the change in chemical shift. And what you see is that there is this very narrow region which shows very high chemical shift perturbation. Okay, which means that interaction is highly specific to those 6 residues, Y, P, W, M, K, R. Only these 6 residues are the ones which are interacting with this partner. And it turns out that these 6 residues are exactly the same as these 6 residues. So we found this in the free protein. There was no partner protein, no DNA, right? And we found that there is this rigid segment, and there is this rigid segment, and there is this flexible region. And then when we did the titration, we found that it is this rigid segment which is binding highly specifically to the protein, right? So that from that we are basically saying that these limbs are functional units in disordered sequences, and most probably these limbs are these small segments which are relatively more rigid compared to the rest of the disordered part. So it turns out that the human proteome has almost 1 million such small motifs. And till date, only 4,000 of those motifs have been discovered. So there is a lot of huge number of such motifs that are yet to be discovered. But the problem is that it's very difficult to identify these small motifs because these are in disordered regions. Their sequence similarity is very poor. These disordered regions get mutated very fast. So bioinformatics is not something that works really well for disordered regions. Also, the binding affinity of these small motifs to the partner proteins are very weak, which means that if someone does a blind pull down assay, that is also not going to work. So we say that this that Nmr Dynamics is something that can be very useful to identify potential slips. So what Nmr Dynamics will do is identify these rigid segments. Now, it's not possible that all rigid segments will turn out to be slims, but some of them can be. So that is a good starting point to look at. So we also try to understand the physical basis of why these small sequences are more rigid compared to others. And for that, we did 200 nanosecond MD simulation. So this was done in 2018. So at that time, this CHARM 36M force field came out, which was designed for disordered regions. But again, I was not very happy with the result that we got. But overall, it seems like we can understand what is happening. So again, the disordered, the flexible part is shown here, and the rigid parts are shown here. And what we can see is that residues in the rigid part, they are actually sampling less conformational space compared to residues which are in the flexible part. Okay. So what we did here is we took all the structures from the MD trajectory and calculated the phi and psi torsion angles and plotted them in this Ramachandran plot. Right. So if you see something is occupying more space in the Ramachandran plot, you can qualitatively say that yes, it is more flexible compared to something which is occupying less space. Right. So from that, it turns out that these two regions have certain characteristics. The residues in this region are hydrophobic, so they are sort of sticking to each other. Okay, but again, there is no structure that is formed. Right, this hydrophobic interaction is restricting the conformational sample. So I would like to put it that way. And this other region is electrostatic. So I'll show you. You can see the sequence here. There is a glutamic acid, lysine, arginine, another arginine. So there is a lot of electrostatic interaction, which means that they are also restricting this conformational sampling. And in this middle part, they are mostly uncharged polar residues. So which means that they are very happy to interact with water, and they are also sampling a lot more conformations. So To explore this more, what we decided is that we thought that, okay, is it possible for us to make this flexible region more rigid? So we are saying that this is rigid because it has this hydrophobic residues, and we are saying that this is rigid because it has these charges, right? So positive charge, negative charge, right? So what if we introduce charges in this region? Will this become relatively more rigid? So that is what we did. We mutated residues so that we introduce this alternate positive and negative charge. So this was one design, and this was another design. So you can see alternate plus and minus charges. And then we went back and did the same NMR experiment. So this is for the wild type. The rigidity is plotted here for each residue. We are showing. I'm showing only the disordered part, right? For the first mutation, where we introduce these two charges, so they are shown by red. You can see the exactly where we introduce the mutation, it goes up. So goes up means it becomes more rigid. So by introducing this plus and minus charges, it actually became more rigid. And then for the third one also, so this introduced another positive charge, it also became more rigid. Right. So the overall rigidity of these two mutants actually went up compared to the wild type. So that's good. So it means that yes, by having this alternate plus and minus charges, a disordered region can become more rigid, relatively more rigid. Now it turns out that this region is actually quite far from the DNA binding. So the DNA binding domain will be somewhere here, right? So this is quite far from that. So my prediction was that these mutations should not make any difference to the actual DNA binding. Snigdha, she went ahead and made these mutants and did the DNA binding. So this is something that we use. We call it electrophoretic mobility shift assay. So here the DNA is actually labeled with a fluorescent dye. We add the protein and run it on a native page. You can see the free DNA here, and when the DNA binds to the protein DNA complex shows up here. We typically quantify the free band, and from that we get the KD. So she measured the KD of the wild type protein and the two mutants, right? And it turns out that the mutants actually become weaker in binding. So even though these mutations that we have introduced is quite far, almost 10 residues, 15 residues far from the DNA binding site, the DNA binding affinity actually becomes weaker, or the KD increases. Right? So structurally, it shows up somewhere here. So that's the DNA binding part. This is the disordered region, and this is where the mutations have been designed. So this is from a crystal structure. You can see that there is a gap here. The reason it's gap because there was no electron density. It is so flexible that this does not show up. Okay, and that is where we have introduced the mutation. So now what we are trying to figure out is why this. So this region obviously interacts with the DNA, and that is why it is getting this affinity is changing. So now we are trying to figure out what exactly is happening there. So this is sort of the summary of what we have seen so far, is that Nmr Dynamics can be used in a very robust manner to identify slips. Right? And we are now applying the same ideas to this human Hox proteins, which are important in leukemia. Okay. So that was what we have done for a disordered protein, right? NMR spectroscopy is, of course, very conducible for folded proteins. So in this example, what I will show you is what we are doing for a folded protein, which is actually an enzyme. So this is a very interesting enzyme. This is the enzyme. It's called Intim. This is the full protein. Okay. Now this protein 1 and protein 2, these sequences do not matter. What this enzyme does is it cleaves here, so it will cut here, and it will also cut here, it itself goes out and it joins these two peptide sequences, right, like this. So this is a complete protein now. This goes out. So this is very similar to RNA splicing. If you have seen RNA splicing, this is very similar to that, but this is happening in the context of proteins, right? And it turns out that this Intin enzyme, because it can do such protein splicing, it has turned out to be a very useful biological tool, biotechnological protein, right? So I'll show you some applications in the last two slides. So this is the reaction mechanism. There are 3 catalytic residues. The first step is cleaving the first peptide bond, formation of this thioester intermediate. Second step is this gets transferred to the last cysteine. So this is a branched intermediate. And at this point, the asparagine cleaves the following peptide bond. So the Intin goes out, right? And then this branched intermediate gets resolved into a linear peptide. So what we are trying to do is we are trying to understand what is so special in this structure that this cysteine cleaves the previous bond. This cysteine takes it from here, and this asparagine cleaves the following peptide bond. Okay, and why these steps happen in stepwise manner. So why this does not happen first? Because if this happens first, then it will just cleave, and what we will get is these two. So if it cleaves here, we will get these two, no spliced product, right? So we will only get C-terminal cleavage. Okay. So there is some driving mechanism that these steps happen in a concerted fashion, one after another. So can we figure that out from the structure and dynamics of this protein? So we went ahead and solved the structure of this protein by NMR spectroscopy. You can see that it looks like this. So it has this donut shape, right? And the protein actually looks like this. The two cysteines, the N terminus and the C terminus, they come out through the middle of the protein like this. So it's a ring structure, and then the two cysteines actually come out from the middle like this. And that is the active site. So that's shown here. The two cysteines are right next to each other, and that's the other catalytic residue. So these are the 3 catalytic residues. So the first reaction happens here, then the peptide gets handed over to here, and then this gets cleaved. So we get this protein 1, protein 2, and the Intin comes out. So we have done also NMR dynamics, and we saw some interesting features, right? I'm not going to go into those. We also did MD simulation, and it seems like maybe it's going through 3 different conformations in the MD simulation. So when I saw that, I was sort of little excited that whether those 3 conformations correspond to these 3 steps, right? Again, we are not sure even now whether it's those 3, whether that correspondence is true or not. So we are trying to figure that out. So to do that, we have to actually design an enzyme assay. Okay. So to do that, what we have done is we have made this big protein. So that's our enzyme. We added ubiquitin in the N terminus and this homeodomain towards the C terminus. There is a his, so it's a 6 histidine, and this is a flag tag. So these are 2 tags that we can detect using Western blotting. Now, potentially, this whole construct can undergo these reactions. If it undergoes the proper splicing reaction, the Intin goes out, and we get the spliced product. We know its molecular weight. It can also undergo N-terminal cleavage. So this is a futile product, right? So it's a side product, not something that is desired. C-terminal cleavage will give something like this, and then these two can also undergo C-terminal cleavage and N-terminal cleavage to give this. Right? So all of these products are actually possible, and we can detect them because we have this his tag in the N-terminus and flag tag at the C-terminus. So what we have done is this is for the wild type protein. So you grow this protein with the bacteria, right? And then you break the bacteria open and do this. So this is staining by Kumasi. So we see all the proteins. This is staining by the his blot. So we are detecting this, right? So these parts. This is staining by the flag tag. So we will detect the full length, this one, this fragment, and this fragment. Okay. So if you see, we do not see the full length, which means that the moment this is synthesized inside the cell, it actually undergoes the splicing reaction. Okay. So at no point of time, so these are 10 minutes, 20 minutes, 1 hour, at no time point, we see the precursor protein. So it's a very fast enzyme. The moment this is synthesized, it undergoes the reaction. We do see this major product is this, the splice product, which is the desired product for this enzyme. But we also see some of the N-terminus and C-terminus cleavage products. We can detect that because of these two flags. So we use this assay to figure out what are the other residues which are involved in this process. So this is just one example. Here we, from the MD simulation and also from literature, we found this aspartic acid to be something that is involved in the first two steps. Right? How can you say it's involved in the first two steps? When we mutate that, there is no spliced product. What we see in the wild type, it's not there. We don't see the precursor in the wild type, we see the precursor in the wild type. And what we see is the C-terminal cleavage. So cleavage at this point. So we can see the C-terminal cleavage product. So this is these first three. Okay. It means that when we mutate this, it is not preventing the C-terminal cleavage, meaning that it is not preventing this step, but it is preventing these two steps. Right. So that way, we have gone through different mutations and figured out whether that residue is involved in these two steps or in the third step. And then again, going back to MD, we looked at whether these residues are N-aspartic acid. This is the catalytic cysteine and the histidine. This is catalytic cysteine and another residue, threonine. So they are energetically coupled, and that coupling undergoes this change at a particular time point. Okay. So when we go back and look at the structure, we do see that there is a conformational change in these side chains, and that change happens simultaneously at exactly this time point for all these 4 amino acids. Okay. So it means that they are forming some sort of cluster that they are helping in switching from one state to another state. Now the question is, what are these two states, and whether this state is involved in this reaction or this state is involved in this reaction. So that is something that we are trying to figure out now. So based on that, what we have done is we have made connections that which are the residues which are connected to each other energetically from the MD simulation and as well as this functional assay that we have done. And we try to put them as these are the residues which are involved in the first two steps, and these are the residues which are involved in the third step. So now this is something that will help us also in the application part that we are trying to do. Okay. So this the main motivation is to figure out this structural basis of the mechanism, but the ulterior motivation is to design this enzyme so that we can do certain things. So what are those certain things? One is to design this self-cleaving soluble tag. Right. So it turns out that so what we want to do is actually make peptides in bacteria. Okay. So synthesis of peptide is something that is quite costly. If we can make it in bacteria, then it becomes quite cheap. But peptides, since they do not have any structure, if you express them in bacteria, they will get cleaved, chopped up very quickly. Right? One way to prevent that is to put some big protein attached to this peptide so that this peptide will not get degraded very fast. So this Intin enzyme, it is a very robust enzyme that can serve as a solubility tag. But this enzyme also has this cleaving property. So it can cleave at its N-terminus or it can cleave at its C-terminus. So what we are trying to do is we are trying to design this Intin enzyme as a solubility tag which can cleave itself out. So this is just an example. This is something that we can use to purify the peptide. That's the enzyme, and that's the peptide. It's a therapeutic peptide. After you purify it, you trigger this enzyme so that it will cut here, and you have this dipeptide, right? So that's the whole step that we can do. So that's one example. The other example is to design split enzyme, meaning that so this is not something that has not been done. So it has been done, and we are also trying to do it with this particular enzyme that we are working with. So if you can split this enzyme, then what happens is that you can use that one to make cyclic peptides inside the cell. And cyclic peptides are again therapeutically very useful because they do not get proteolyzed as quickly as linear peptides. And they can also be used to synthesize proteins with modification, chemical modification. So suppose I want to make a protein which has some unnatural amino acid. So in that case, what I will do is I have this split enzyme, right? So this is my protein. I fuse it to this enzyme, and this is the other part of the enzyme, and a small peptide which has an unnatural amino acid or some other chemical modification. So this can be chemically synthesized, and this can be produced in bacteria. When you put them together, these two come together, and they will reconstitute the functional enzyme. It cleaves itself out, and it joins these two. So now you have a protein with this unnatural amino acid or a phosphorylation or any other chemical modification that you want in a very specific site. Okay. So this is something that we are trying to do now. We are trying to design split enzymes that can be used for making this type of proteins with unnatural amino acids, and also make this type of cyclic peptide library inside the bacteria. So I have a lot of collaboration, so I like to collaborate with lots of people, right? So there are many people in IIT Kharagpur and also outside IIT, and these are the funding agencies. Ultimately, you need money to do research, right? So, and these are just the conclusions that I have just mentioned. Yeah, I'll be happy to take questions.
Thank you, Dr. Dev. So we are now open for questions. Any questions? And those who are online, can you please raise your hand, and then we'll take up those questions later. Yeah, they'll be there. So, yeah.
Right.
Apart from simulation, MD simulation, it is also better.
So we did from both NMR and MD. So this one is from MD. Okay. So, so this is something that again, I'm not sure if it is right or wrong, but this is one way of doing it. So one problem with this type of protein. So you see that this is the folded protein, right? This is the folded part, and this is the disordered part. If we do RMSF, right, in that case, what happens is that this part will look very similar to this, but here it keeps on increasing like this. So as something goes further from the folded part, its flexibility keeps on increasing because you have this type of motion, right? So anything that is far will have seems like it has more motion than something that is close. So it was not very useful for us. So for that, what we did was we actually went to Ramachandran Plot, because here in Ramachandran Plot is actually, I mean, it's a for structural biologists, it's most probably the most useful thing, right? Because any conformation, no matter what conformation you can think, can be represented in this bound manner, right? So all conformations, all combinations of phi and psi show up in this. Right? So what we did was for every residue, we calculated the phi and psi torsion angle from this 200,000 structures, right, from the MD trajectory. So for this particular residue, those are plotted here. Right? And then we divided this Ramachandran plot into these 2x2 grid boxes. Okay. And then you count how many grid boxes are occupied. And So then the total number of grid boxes we know, and the number of boxes that are occupied. So you divide that by the total, and you get a number. So that's the flexibility index. So if more grid boxes are occupied, then the flexibility goes up. If less number of grid boxes are occupied, then the flexibility is. So that sort of tallied well with the NMR data that we have seen. Like this.
Just as a follow-up comment, whatever you have done is like counting the grid. There is a formal way of defining conformational entropy, which you have heard probably about Shannon entropy.
Right.
So they are actually, if you have a probability distribution here, histogram.
Right.
So the whole information can be, there is an integral. So if since you already have this histogram, there is an expression that you can, this can be integrated to make, and then you can have a conformational entropy of that ith vessel.
Right. So that is what we wanted to do later on. I mean, in this case, we did
I think this is also will give a kind of a similar trend.
Yes, but it's not taking into account that these regions have more population than. That is what you are saying, right?
Essentially, it will have a weighted average.
Average.
It's like a count, but right now you are this also you are counting on, and this also you are counting on. But it will take a weightage of the probability distribution itself, and then it will give an estimate of the entropy. Right. So essentially, conformational.
So that is exactly what we want.
Exactly.
Okay.
You can just look up this Shannon entropy. There is an expression that people use it normally to compute conformational entropy of protein side chains. Okay. So you have multiple rotamers, let's say.
This is
and
similar to that.
Yeah. So if you know the population of different rotamers, so it is the sum of Pi, ln Pi. So Pi is the probability of the ith rotamer, something like that. But I think that is a more rigorous expression. Right. But this also gives similar trend. Yeah.
You have mentioned that pretty charge. It is the polar, non-polar residue by charge residue. The rigidity,
right?
Formation of salt bridges with those charge residues.
Yes, yes. So when we are introducing these charges, right? So then what happens is that the number of conformations that are sampled are restricted. I mean, it can happen both ways, either by attraction or by repulsion. But ultimately, the number of conformations that are sampled are restricted.
Question?
Yeah. For converting like correlating with J NOE, so you have one like 3 factor kind of 0.8 something.
Sorry, let me go to that. This one?
Like this one, yeah.
Oh, this 0.87. Yeah. So what is that? Okay, so yes, sorry, I actually sort of glossed over that. So it turns out that this R1, R2, and NOE, these are related to 5 spectral densities. Okay, so those 5 spectral densities are given as J0, J omega n, J omega h. So omega n is the Larmor frequency of nitrogen. We are looking at M15 nitrogen, and omega h is the proton, 1h. And then 2 more factors come in, which is omega h minus omega n and omega h plus omega n. So there are 5 terms, but we have only 3 experimental values. So if we want to solve 5 spectral densities from 3, we cannot do that. Right? So later on, what was found that you can actually combine these 3 higher frequency terms, the j omega h minus omega n, j omega h plus j omega n, and j omega h, these 3. You can combine those 3 into this one single term. Okay. So that is why this 0.87 omega h comes. So it's basically an approximation of those 3 terms together. So now what you have is 3 and 3. So from these 3 experimental parameters, you can calculate these 3 spectral densities.
Like some of your part, like protein part is missing. So the residue. Yeah. Yes. So you muted this, right?
So okay, so this is actually from a crystal structure, right? So in crystal structure, what happens is if something is highly flexible, then you don't get any electron density for those. So that's why it shows up as missing. But when we are doing NMR, we can see everything, right? So for NMR, it doesn't matter whether it is rigid or flexible.
Okay.
We can see it. So this is from the crystal structure, right? So that is what I was trying to. Maybe I was not very clear. So the point was that the mutation that we made is in this site. So if you go to the crystal structure, you don't get any information because it's missing. So we, from the structure, it's not clear why making mutation in this site gives this change. Yes.
Spectral density part and J value it be directly from the experiment because J is some kind of function.
Yes. Yes. So we can actually calculate that directly from experiment. So spectral density function is your omega square tau c by 1 plus omega square, something like that, 2 by 5, right? So that omega is this. Okay. So but then again, there is a expression for that. So there is a expression for how this J0, J omega n, and J omega h, this one, are related to these three. So if we know these three, we can actually calculate these three. From that, we can calculate these three, the spectral density value at these three frequencies.
Maybe let's ask online. Let's see. Yes. Is there any question from those who are online? I don't see any raised hand. Is there any question? Okay, I guess if not, is there any other question? Yeah.
Yes.
Like you say that in the titrated, I don't know about the details of the experiment, but Fox protein with its partner protein, right? Without DNA.
Right.
And you have like identified some rigid regions which are actually interacting with its identified some rigid regions which are actually interacting with its partner.
Correct.
So, like how it will change when you have DNA also, because when you introduce charge residues, it gives us more rigidity because of the interaction. So, like I just want to know that if there is any kind of allosteric effect that which part is interacting with DNA, if that interaction can change the.
Yes, so we have actually tried to look at that. Let me see if I can. Maybe this one. So again, this is a cartoon diagram, but it sort of shows what happens when the DNA is there. So these two double lines are represent the DNA, double-stranded DNA. Okay. So this protein and this protein both have DNA binding domain. So they bind to DNA by themselves, but they interact with each other through this disordered part. Okay. So this disordered part is not interacting much with the DNA, but this disordered part is needed for the interaction of these two proteins. Okay. Now, to answer your question, what we are trying to do now is actually look at these complex, all three at the same time, and also look at the wild type versus the mutant. So that we are doing using this M-site screen. So because we can do that in the presence of DNA. So that we have done. The results are actually not very encouraging because this affinity is actually quite weak. But the binding affinity of the protein to the DNA is quite tight. So there is 3 orders of magnitude difference between them. Okay. So the MSI experiment is actually not working quite well. So we have to figure out some other way of looking at that. But yes, that is something that will be the next thing.
Okay. If there are no more questions, let us thank Dr. Dev.
Thank you.
Hope there is tea outside. So. না মাস্টারটাকে বন্ধ করে দিলে ডিলিট করে দিতে চাই লাস্ট করে দিতে পারো
And I'm going to get off the